Good morning, everyone, and welcome. I'm Dr. S.K. Singh, and I'm glad to have you here today. We'll be discussing extractables and leachables studies, a crucial aspect of ensuring pharmaceutical safety and quality. In this presentation, I'll walk you through the key concepts, methodologies, and importance of these studies. So, let's get started and explore how these processes help us protect patients and meet regulatory standards. Let's start by discussing extractables and leachables, often abbreviated as E and L studies. These are scientific evaluations that play a crucial role in our industry. They are performed to identify, characterize, and assess chemical substances that might migrate from various materials used in drug manufacturing. Specifically, these substances can come from container closure systems, packaging materials, processing equipment, or even medical devices. The goal is to understand what chemicals could potentially migrate into the drug product under certain conditions. Now, why is this important? Well, the main objectives of E and L studies are to ensure patient safety, maintain the quality of the drug product, and demonstrate compliance with regulatory requirements. Additionally, these studies support a risk-based approach to decision-making, helping us identify and mitigate potential risks early in the development process. Overall, E and L studies are a fundamental part of ensuring that the medicines we develop are safe, effective, and compliant with all necessary regulations. Let's talk about extractables. These are chemical compounds that can be forcibly pulled out of materials under certain conditions. Imagine exposing a material to strong solvents, high temperatures, or prolonged exposure. These conditions can cause certain chemicals to leach out. Understanding extractables is crucial because it helps us identify what chemicals might come out of our materials under stress. This knowledge allows us to predict potential leachables, which are the substances that could migrate into products or the environment. By studying extractables, we can better evaluate the safety and compatibility of our materials, supporting informed material selection and risk assessment. Typically, we focus on this during key phases like development, when we're qualifying packaging, or when there's a change in suppliers or new materials are introduced. This process ensures that we maintain product safety and quality throughout the life cycle. Overall, understanding extractables plays a vital role in safeguarding both our products and consumers. Let's talk about leachables. These are chemical compounds that can actually migrate from the materials used in packaging or containers into the drug product itself. This process happens under normal storage conditions and throughout the product's shelf life. It's important to understand that these compounds can potentially impact patient safety, so evaluating them is a key part of our safety assessments. The main purpose of studying leachables is to ensure that the drug remains safe for patients. We also want to confirm that the product maintains its quality and complies with regulatory standards. This helps us demonstrate that everything is within acceptable limits and that the product is reliable. Typically, we study leachables during stability studies, which simulate how the product will be stored over time. We look at commercial storage conditions, which are the normal environments where the product will be kept. And of course, we perform shelf life assessments to understand how leachables might change or increase over the product's entire lifespan. All of these efforts are crucial in supporting regulatory submissions, ensuring that the product is safe, effective, and compliant throughout its intended use. So, by thoroughly evaluating leachables, we are safeguarding both the product's quality and the health of the patients who use it. Let's take a moment to look at the risk management process. This is a crucial part of any project or organization because it helps us identify, assess, and address potential risks before they become bigger problems. The process typically starts with identifying risks, so we ask ourselves, what could possibly go wrong? Once we've identified those risks, we move on to assessing their likelihood and potential impact. This helps us prioritize which risks need our immediate attention. After that, we develop strategies to either mitigate, transfer, or accept those risks, depending on their severity. And finally, we implement those strategies and continuously monitor the risks throughout the project lifecycle. This ongoing process ensures we're proactive rather than reactive, and it ties directly into our overall goal of maintaining stability and success in our projects. Let's dive into assessment, related risk identification and mitigation, specifically for manufacturing components and systems. This is a critical area because identifying potential risks early on allows us to prevent costly failures down the line. The first key point is that risk identification involves a thorough analysis of all stages in the manufacturing process. We need to look at every component, every system, and every process to spot where things might go wrong. Once we've identified these risks, the next step is mitigation. This means putting strategies in place to reduce or eliminate those risks. It could involve design changes, process improvements, or additional quality checks. What's important here is that this risk management approach isn't a one-time task. It's an ongoing process. As we develop and refine our manufacturing systems, new risks can emerge, so continuous assessment is essential. Connecting this to the overall theme, effective risk identification and mitigation are fundamental to ensuring the reliability and safety of our manufacturing components. It helps us maintain high standards, reduce downtime, and ultimately deliver better products to our customers. Let's take a look at this example to understand how testing is conducted for rubber stoppers used with injectable vials. The scenario here involves evaluating the stopper's performance under specific conditions. We test the stopper's compatibility with different solvents, namely water, isopropanol or IPA, and hexane. These solvents are chosen because they represent various chemical environments the stopper might encounter in real-world use. Next, we consider the temperature condition, which is set at 70 degrees Celsius. Elevated temperatures like this help us simulate accelerated aging processes, giving us insights into how the rubber stopper might behave over time. And finally, the duration of the test is 24 hours. This time frame allows us to observe any potential degradation, swelling, or other changes in the material when exposed to these solvents under the specified temperature. Overall, this example illustrates the importance of testing under controlled, but challenging conditions to ensure the integrity, compatibility, and safety of rubber stoppers in pharmaceutical applications. In this slide, we're looking at the real storage study, which gives us practical insights into how our product performs under typical conditions. The study focused on an injectable product, which, as you know, is a common form for many therapies. We tested storage at 2 different temperatures, 5 degrees Celsius, which is standard refrigeration, and 25 degrees Celsius with 60% relative humidity, simulating room temperature conditions. We monitored the product over a period of up to 24 months to assess stability and safety over time. One of the key aspects we're examining here is the detection of leachables. These are trace substances that might migrate from packaging or container components into the product. Identifying and understanding these leachables is crucial because they can impact product quality and safety. Throughout this study, we closely analyzed these potential contaminants to ensure they remain within acceptable limits, maintaining the integrity of the product throughout its shelf life. Overall, this real storage study helps us confirm that under these conditions, our injectable remains stable and safe for up to 2 years, which is vital for clinical and commercial use. It's a critical piece of the puzzle in ensuring our product's reliability and safety for patients. All right, let's dive into slide 9. First, we'll look at the safety threshold, which is based on the PDE or permitted daily exposure, following regulatory guidance. This helps us establish a safe limit for exposure levels. Next, we have the daily exposure calculation. The formula is quite straightforward. Exposure in parts per billion, or ppb, equals the measured ppb times the daily dose in milliliters, divided by 1. For example, if we measure a certain contaminant at a specific ppb level, and the daily dose is known, we can calculate the actual exposure to determine if it stays within safe limits. This calculation is essential because it allows us to assess risk based on real-world usage. So the key takeaway here is understanding how to translate measured contaminant levels into actual exposure, which we compare against the safety thresholds. This helps us ensure that our practices remain within the acceptable safety margins. Now, let's take a closer look at systemic and local toxicity thresholds. These thresholds are crucial because they help us understand the levels at which a substance can cause harm, either throughout the whole body or in specific areas. When we talk about systemic toxicity, we're referring to the overall impact on the body. How much of a substance can be tolerated before it starts to cause adverse effects that affect multiple organs or systems? On the other hand, local toxicity thresholds focus on specific tissues or sites, like the skin or respiratory tract, where the substance might cause localized damage or irritation. Recognizing these thresholds allows us to establish safe exposure limits and design better safety protocols. It's important because, as we've seen earlier, managing both systemic and local risks is key to ensuring safety in our applications. Understanding these thresholds ultimately helps us protect health while still achieving our desired outcomes with these materials. So, why are USB 1663 and 1664 important? Well, they provide standardized approaches to designing, performing, and interpreting studies, which really helps ensure consistency and reliability across different laboratories and testing methods. This standardization is crucial for maintaining quality and trust in the results we obtain. There's also a strong emphasis on safety-based limits, meaning these guidelines focus on protecting public health by setting clear, scientifically backed thresholds for various substances. When it comes to analytical techniques, these standards cover a range of methods suited for different types of analytes. For example, GCMS is used for detecting volatile organic compounds, while LC-MS is particularly effective for semi-volatile compounds. For metals, ICP-MS is the go-to technique because of its sensitivity and precision. Additionally, Headspace GC is highlighted as a key technique for analyzing samples where volatile compounds are involved, especially in complex matrices. Overall, these standards and techniques help us ensure that our studies are robust, reproducible, and aligned with safety concerns, which ultimately supports the integrity of our data and the protection of public health. Let's talk about how we interpret and assess the risks associated with leachable substances. First, we ask, does the leachable level exceed the PDE or permissible daily exposure? If it does, that's a clear indicator that we need to pay closer attention. Next, we consider whether the compound is structurally toxic. Specifically, is it mutagenic or an endocrine disruptor? These factors are crucial because they can impact health even at low levels. We also look at the trends over time. Are the levels increasing? If they are, that might signal ongoing issues that require action. Based on this assessment, we make risk decisions. If the data is within safe limits, we might accept the current situation, indicated by the checkmark. If there are uncertainties or potential concerns, further investigation is warranted, represented by the warning symbol. And if the risk is significant, we should consider mitigation strategies or changing the material altogether, marked by the cross. Understanding these key questions helps us make informed, responsible decisions to ensure safety and compliance throughout our processes. Let's take a closer look at the overview on aspects to consider for the risk matrix. As you can see, we've included some key abbreviations here, such as CSF for cerebrospinal fluid, DP for drug product, IM for intramuscular, IV for intravenous, and SC for subcutaneous. These are important factors because they influence the risk profile of different formulations and delivery methods. When we talk about interpretation and risk, especially in this ongoing discussion, it's crucial to understand how these elements interact. For example, the route of administration, whether it's IM, IV, or SC, can significantly impact the safety and efficacy profile of a drug. Likewise, considering the specific characteristics of the drug product itself, including its formulation and stability, is vital. In developing a risk matrix, we need to evaluate each of these aspects carefully. Think about how the cerebrospinal fluid might affect drug distribution or how certain delivery routes might introduce specific risks. By systematically analyzing these factors, we can better predict potential issues and prioritize our mitigation strategies. Overall, the goal here is to ensure we have a comprehensive understanding of all relevant aspects to effectively manage risks throughout the development and manufacturing processes. This approach helps us make informed decisions, ultimately leading to safer and more effective therapies. Let's wrap up with some key takeaways. E&L studies are truly essential for ensuring patient safety and maintaining compliance throughout the product lifecycle. Starting with a thorough extractables profiling gives us a clear understanding of what chemicals could potentially leach out. From there, using a risk assessment helps us determine where to focus our leachables testing, making the process more efficient and targeted. It's also crucial to align our results with global guidance standards, such as those from USB, ICH, FDA, and EMA. This alignment not only ensures regulatory compliance, but also supports the overall safety and quality of our products. Remember, a solid foundation in extractables profiling, guided by risk assessment, and aligned with international standards, is the best approach to managing leachables effectively. This comprehensive strategy ultimately safeguards patients and upholds our commitment to high-quality products. For further support, please don't hesitate to reach out. If you have any questions or need additional information, I'm here to help. You can contact me through the provided details. Thank you so much for your time and attention today. I really appreciate your engagement and hope you found this presentation valuable. Remember, ongoing support is just a message away, so feel free to get in touch anytime. Thanks again, and have a great day.